Hi, everyone. Good afternoon. Everybody seeing this okay?
Yes.
Okay. Yeah, so it's 7 p.m. here and it is good to see familiar faces. So magandang umaga po sa lahat na nasa Pilipinas at sa karatig bansa. So today I will be sharing Seaweeds for human health applications. So a brief background, I'm in Oregon State. Those are the programs that we're good at, just so. And so I'm part of the Department of Food Science and Technology. It's one of the oldest in the country. And the good thing is we do specialize in specific aspects of the industry. We have good support from the dairy, the fermentation field. We have a lot of beer, wine, mead in the department. So it goes hand in hand with a lot of cheese. We do have a good food system, sustainability, and safety programs going on. And I'm part of the seafood lab, so that's in a different campus. So yeah, so the Department of Food Science has 3 major locations in Oregon. So we have the Wiegand Hall, which is in the campus, in the main academic campus. And then we have the Food Innovation Center, which is in Portland, Oregon, where most of the industry consults for product development and improvement are being conducted. And then we have the Seafood Research Education Center that's the main part of the department that I'm a part of, that is in Astoria, which is the coast. And I am in a totally different building. I am in the Linus Pauling Center. So kahit po meron po kaming location sa department, ako po ay nasa kabilang building, which is kind of cool because the building itself is named after Linus Pauling. He's a Nobel Prize researcher working with vitamin C. I guess we have an accident. But yeah, so that's one of the things that is advantageous because the building itself is housing a lot of researches, mainly on vitamin E, vitamin D, a lot of micronutrients, supplementation research, both in animal and human studies. So it's very advantageous when we talk about seminar access and free food, of course, pre-COVID. So yeah. So since Nilo asked me to kind of walk you through my academic journey as a student and as a researcher, so I'll share some snippets of the aspects of my life here. So I started in 2017 with the Fulbright scholarship, and so I'm so sold out to going and out there finding answers, discovering, and meeting challenges. But on a regular basis, this is really the real deal. You get to have confrontational conversations, you and yourself, what's going to be next. So yeah. So today's presentation, going back in track, I will walk you through these 3 major things that I would want to cover. So mainly seaweeds as food, how do we get there? Meaning how do we reach to a point where like we could gain support and traction that we could go further in our research, and the prospects as to where we could begin at this point. So seaweeds in general, macroalgae, there's a globally recognized importance of seaweed as a component in a diet and a functional food source, given that it has been anecdotally incorporated in the diet, and with major seaweed consuming communities having longer lifespan and lesser disease incidence. However, there's also a consensus that there has to be more efforts towards understanding the bioavailability of whatever macromolecules that we're looking at, minerals and vitamins that we suspect to be bioactive. And so there's that challenge that needs to be confronted. And so, but then again, that does not close the door that seaweed is a promising functional food source. Here I'm showing you major components of a dietary seaweed and what we're talking when it comes to like supplementation. What are the major macromolecules at play? So you can see here we are talking about mainly fiber, at some point protein for red seaweed mainly, and then some of the ash minerals. We do recognize a lot of work with seaweed salt substitutions. And of course, you know your algal polysaccharides. We do have a big market for that in the country. Now, dietary seaweeds in the context of obesity has been quite solid as a research ground. Since 1980, dietary polysaccharides from seaweeds has been established to, one, have hypolipidemic effects, meaning it has the ability of reducing blood lipids, be it triglycerides or cholesterol. However, what really gained traction was the effect of the polysaccharide sodium alginate in inducing satiety, meaning busog ka, you don't feel as much hunger, and so there in having that, you don't tend to crave for food, and so you have less caloric intake in the process. Another aspect to that is that because it has a gelling capacity, your nutrient absorption in your gut is somewhat delayed, and so there's a gradual introduction of blood glucose in your system, and so that is protective as well. And so this particular paper covers most of the clinical trials on alginate, and if you're interested, you can look into it. An emerging application for seaweeds right now is for prebiotic application. So that means your algal polysaccharides that are not available to the body, they play as more like a fiber. And so in this context, you could imagine how different kinds of algal polysaccharides could be treated and processed by the body and how that would benefit. And so here, this is from a very recent paper. It's more of a perspective. And so you could see how this three major algal sources, we have green, red, and brown, polysaccharides coming from these different sources could have different metabolic fates in the lumen. And so why does this matter? It matters because if you look at it from the standpoint of fiber, like everybody says, like, oh, fiber would be good, but there has to be a certain caveat to that because not all fiber are created equal. And so if you look at the context of soluble fiber, that could somehow help in delaying a gastric transit versus your insoluble fiber that would just absorb water and just like facilitate that bowel movement. And so those are two different things. And so particularly for this one, what they were pointing out was based on in vitro and in vivo studies, at least they included some of the rodent and pig studies. They knew that agar and carrageenan, alginate and laminarin have prebiotic effect in specific bacterial communities that are preferred and are usual members of the healthy gut community. And so in the process, since they are also able to be metabolized, they could yield short-chain fatty acids that could help improve colonic health, especially addressing colonocytes. And so that is beneficial. However, there's a contention right now with sulfatate polysaccharides, polysaccharides, that there are chances that they could actually affect mitochondrial functions. And so there's like a big research area, burgeoning area at that point in making sure like how this sulfatate polysaccharides are actually being handled, since there are specific bacterial species that could take away that free sulfate and utilize that. And so if they proliferate, that could not be as beneficial as one would expect. So there's like a balance to that. So another thing other than polysaccharides and fiber, we do have proteins from the seaweed. As I've shown earlier, there's a significant chunk of protein that you could actually get from seaweed. However, there's a challenge to that because seaweed are rich in polyphenols as well. So bioavailability wise, the protein from seaweeds are not as readily accessible. And so, but anyway, from the protein standpoint, what has been growing in the research field is you actually work with the digested protein from the seaweed, and then from there you are working with potentially bioactive peptides, since they're like chopped off pieces of their substrate protein. And so there has been a lot of reports for in vitro work on antihypertensive, antioxidant, anti-diabetic peptides from seaweed protein. And I'm showing you this proposed mechanism for bioactive peptides, and this is across the board, not just seaweed, but these are proposed mechanisms. But I would like to point out that the same mechanism are also viewed in the context of small molecules, say polyphenols from seaweeds. There has been a lot of work on fluorotannins, echols. Those small molecules have established bioactivity in vitro and in vivo, at least for rodent models. And then for like carotenoids like glucoxanthin, the same mechanisms are also recognized meeting these challenges. So first, let's look at this anti-diabetic treatment. And so I will show you this pathway here. So bioactive peptides and small molecules could stimulate your insulin receptor. And I'm glad Kuya Brian talked about this earlier. And so essentially in the simulation of your insulin receptor, you could go trigger the cascade for insulin signaling. And so in the process, you have the promotion of the signaling cascade for insulin as well as AMPK here. So not only do you have glycogen synthesis kicking in, ibig sabihin you're storing your energy sources, you're also putting a halt to gluconeogenesis. So your liver, say for example, is not generating glucose substrates, and so you are stopping like another caloric production. And so you're limiting that, and in the process, you don't have glucose floating in your blood. Another side to that is when you promote AMPK signaling, you also upregulate GLUT4, and so GLUT4 is insulin-sensitive transporter for glucose, and so that means yung transport ng glucose in your blood is sped up. And so imagine in the context of diabetes, you have elevated blood glucose, and so you want the fastest way to clear that up. So either you have good insulin signaling that could stop production of your glucose, or like store it up as a glycogen, or just like acquire transporters that would easily clean that up in your blood. And essentially bioactive peptides and some of the small molecules from seaweed has been shown in numerous studies to accomplish this, and thus the anti-diabetic activities that has been reported associated to this. Another thing is the anti-obesity effects. And so the idea with obesity really is that you do have an increased amount of fat mass. And so because you have significant amount of fat mass, one, you have to look at it as a protective mechanism, because you're trying to store up whatever calories floating in your blood in like, say, like an empty bag. But if they increase in the process, they could be inflamed given the stress that they have to accommodate all those excess calories that they have to house. And so that would be stressful. And so most of obese patients graduate in more complicated disease conditions because of metabolic aberrations arising from just having that significantly high amount of fat mass. And so the idea is if we could suppress the accumulation of this significant fat mass, that would be beneficial. And so here, some of the studies would target PPAR gamma. So this is associated with adipocyte differentiation. And so if you have a lot of fat cells, so you could accommodate more of those caloric excess, but in the process, if it does not differentiate healthily, you could be chronically inflamed as well. So there's a balance to that. Another aspect is targeting PPAR gamma. Of PPAR gamma is PPAR alpha, and so that would be upregulating beta oxidation. So kung hindi mo pwedeng bawasan yung mga bags na pwedeng mag-contain, pwedeng i-increase ang energy expenditure. So you are burning your caloric storage. So that's another angle to it. And so I know Caulerpa, polyphenols from Caulerpa has been shown to do that in vivo, in vitro first when they were screening using adipocytes, and then they showed that in vivo using a mouse model. And then the anti-inflammatory, which I am mainly interested with. And so the idea with the anti-inflammatory effects would be halting the three major pathways. And so here, I'm not sure how visible this will be, but essentially, if you prevent IKB degradation, so that is a complex that is outside the cell, if you degrade IKB, it would not be able to migrate on the nucleus, and so inflammatory genes would not be upregulated, and so that would be an anti-inflammatory effect. At the same time, you have the MAPK3 as well as JAK-STAT. And so the main goal is if you suppress the signaling pathways, there could be some degree of rescue, which is important in the context of chronic inflammation. So say, for example, individuals who have sustained level of inflammation. Inflammation itself is good, but if it stays longer, it does not get resolved. It's like, imagine you have a slight fever, but it persists for a long time. And so one, that's annoying. And two, that would be dangerous. Imagine how much aberration that is accomplishing in your system while you feel like, well, this is bearable, but even then that's not your normal state. So yeah, so I've covered fiber, I've covered peptides and some polyphenols. Now, so that is from the side point of seaweed as food. However, there's also a growing work on seaweed drugs. So here, this is from 2014, but they did cover a lot of small molecules from seaweed, mainly enzyme inhibitors. And so they provided major species of seaweeds that this small molecules were obtained from. And so they did some sort of like a summary showing that 49% were like from red algae and 43 from brown, and then fewer from green seaweeds. So an example for that, a very concrete example. So NC2 is a commercially available food supplement. And so what it is it's targeting diabetic patients in how they could regulate blood sugar. And so you can see here, the y-axis is blood sugar in millimoles per liter. And here, this product, by the way, is a polyphenol bleach extract. And so 30 minutes before ingestion of the carbohydrate source, they had the supplement. So you have NC2, and then you have a placebo. And so 30 minutes before they gave this, and then after that, they had the challenge of whole white bread. And so you could see here over time, which is in the x-axis, you could see that blood glucose would increase the moment you administer the food. However, the supplementation of NC2 dampened the increase of blood glucose, which shows some degree of rescue in the excessive levels of blood glucose in the body for diabetic patients, but two, it speeds up the drop. And so the faster you get to your basal level, the better. You don't want blood glucose to stay so long in your blood that you could glycate into your safer blood cells. So essentially, this product claims that their polyphenol extract does have alpha amylase inhibitors as well as alpha glucosidase. Now, those enzymes chop off carbohydrates, and so in the process, if you delay the activity of the introduction of glucose in your system by dampening enzyme activity, that would be protective in ensuring that for diabetic patients, they don't have this like influx of glucose in their blood. So that is a protective thing. Next here is, this here is one of the results for this Alzheimer trial. So this drug called GV971. So this is a sodium oligomenate. This is from Seaweeds. So in China, this has been recently approved. So it's commercially marketed as a drug for Alzheimer's disease. But here in the States, it's still in phase 3, meaning it's on a clinical trial for deceased patients. So meaning they're testing it in like vulnerable populations, mainly for Alzheimer patients. And so here in the x-axis, these are actually Alzheimer disease cognitive scores. So they have an evaluation in their patients. And so you could see here, this is the score of patients getting the placebo and the drug. And so you could see that over time, those who got the drug did have some degree of improvement over this 36-week period. So it's called seaweed sugar, essentially, because it's an oligomenate. So if you feel like wanting to read more on this, the paper is available. This is a multi-center trial, so there's some challenges to that. But even then, the fact that it got through phase 3 here in the States, that says something. Yeah. So another case that we could look at is, say, for example, cancer. It's more of a epidemiology and an intervention story. And this is often the case with traditional medicine. You know, there's like an anecdotal evidence that this thing works. but the question is how. And I think that's one thing that we have to take advantage of, given that we do have a lot of that, especially in Asia, for what our grandparents have been like suggesting that we take, or recommending that this would work, something like that. So here, specifically, they were looking at prevalence and what dietary practices are associated with breast cancer. And so eventually this prior work would lead to this clinical trial which involved seaweed powder, which are concentrated with polyphenol. And so they did this, I think, in 1991. While the rescuing effect for early breast cancer was not as profound, this gave way to more cancer work, at least clinical trial-wise. And so now I know that sequoidin, it's algal polysaccharide, has been used for squamous cell carcinoma in the head and the neck, as well as liver cancer, and in as paired to chemotherapy for non-small lung cancer. So those are so NCTs. These are clinical trial number. These are like identifiers when you look into a library of registered clinical trials, so people can keep track what has been registered, what has been completed, and what is still going on. So we covered polyphenol, we have polysaccharide sequoidin. Next would be calahalide. So this is actually a peptide. So this was discovered in 1993 by Dr. Heyman. He's a PhD, I think, in Hawaii at that time. So they found this in a mollusk that feeds on the seaweed and Boreopsis. And so they isolated it. And as of now, this has a lot of wide application as an anti-tumor treatment. And so if you could look at it, it's associated with a pharma mark, because this a lot of trials is funded by a private industry. It is so costly. And so you could imagine people invest for the most promising ones, because at the end of the tunnel, it would really require a lot. So you need to have strong groundwork. What I'm really interested is on metabolic health applications. So here are some of the metabolic health studies, clinical trials that has been registered using seaweed or derivatives thereof. So that would mean extracts, powder, or purified components like fucoxanthin and fucoidin. And so you could see here, there's a unifying theme for what their therapeutic targets are. So generally, you're looking at glycemic control. As I discussed earlier with NT2, you could imagine most of the bioactive components here targets the drop in blood glucose or increasing insulin sensitivity. And so we are talking about carbohydrates inhibitors, as well as those that could stimulate sensitivity and upregulate some of glucose uptake, for example. Another would be caloric handling. As I mentioned earlier, polysaccharide, especially soluble fiber, would facilitate delay in nutrient absorption. And so one, that could ease out the introduction of glucose in your system, but two, that could also extend the time that you feel full, and so you don't eat a lot. And so that could be helpful when you're like looking into weight loss, or improving even some of your prognosis when you're already diabetic, something like that. And another burgeoning field would be the gut microbiome. So the idea with the gut microbiome targeted approach is that in the context of obesity, mainly triggered by high-fat diet or things related to metabolic aberration, the idea is that your gut microbiome has reached like a dysbiotic space. This means that like from the original profile, it has significantly shifted. And so pathobionts, the unhealthy gut flora, was proliferating. And so because it is proliferating, at that point, your body is not able to compensate. And so this dysbiotic gut microbiome, one, is producing more energy, because you could imagine your gut also processes like fiber, right? So it generates energy in that too, but at the same time, it could allow proliferation of pathobionts that could trigger inflammation in your gut. And so this degree of inflammation could adapt to a chronic inflammatory state. And so the self-perpetuation of this inflammatory state could just keep going on, and so that unhealthy state could just be sustained. So overall, that's the target for the metabolic health clinical trial so far that has been registered. So since this part of the presentation really asks where are we at now, I think the clinical trial would give a better context because one, they kind of filtered through the ton of data available, and so they focus on whatever that has the strongest therapeutic potential, and so they're willing to fund it in clinical studies. And so imagine if you look like in a quick search, there's so many studies that would report in like different bioactivities from seaweed, but the bottleneck really would be the clinical trials because one, it's costly, and two, it would require a lot of controls in doing this. And so they have to make sure that whatever reaches that level would be promising enough and worth the budget. Two, it's a good way to start like a situationer because that shows whatever thing you are testing also has a market niche, and that's why people are willing to fund studies. So imagine this takes a long time, and so you could imagine the process of drug discovery taking a while significantly, but if it does go beyond that, that means it has gone through a lot of filters. So that is protective enough. But how do we get there? So there is in a point where it's worth considering for a clinical trial. It's worth listening a pitch for a clinical trial at least. As I've shown earlier, we have to do our groundwork. We have to go for basic research, non-clinical studies, because at the end of the day, if the groundwork is not solid enough, nobody would be willing to fund a clinical study. I guess unless it's like, okay, we have extra money, we might as well fund your study. Yeah, so in going there, what would be the exploration strategies? So I'm providing 3 examples, at least that I'm familiar with as I'm using this in my study. So you could go for in vivo, in vitro, or in silico. I'd start with in vivo. In 2018, I did my own study. It's a study from our lab that I anchored. And so essentially what we did was we had Palmaria mollis, this is seaweed grown in Pacific Northwest, and then we had Undario pinnatifida. We freeze-dried this one and then incorporated it in our pellet and fed this to our mouse. And so the mouse model that I'm working is an OB mouse. This is C57BL6J mice. And so this mouse is essentially prone to obesity because it does not have a leptin receptor. So, and it does not produce leptin as well. So it's OB. And because it does not have this ability, it cannot help but overfeed. And in the process of overfeeding, of course, you have excess calorie and you cannot help but become obese. And so with this, what we had is we had an 8-week feeding, and we looked into the glucose tolerance, how my mouse were able to handle glucose after fasting. And then we collected tissues and looked into genes and proteins that we could monitor the extent of obesity, like phenotypically what has been changed. So yeah, and for the purpose of this presentation, I'm just showing you the result involving dose, because that's the seaweed that I'm interested with. And so this was published in 2020. So here, this is blood glucose, so as I've shown earlier in NT2. So you could see here the blood glucose over time detected in the blood of the mouse. And so these are fasted mouse. So you would have a fasted mouse and introducing glucose intraperitoneally, and you would routinely check the blood glucose. And so you could see here, well, this is not significantly different for, by the way, the green would be the dose and the red would be the high fat. They're both high fat, but the high fat with dose is just a top off supplement. And so you could see here, one, the drop was instantaneous in 30 minutes, while there was a steady increase still for high fat. And so this trend is almost similar to the low fat. And so we were hoping that, well, maybe there is some subtle anti-diabetic factor that we could probe into. And so this particular project is more of a hypothesis search kind of thing. We also looked into plasma MCP1, so that's monocyte chemotactic protein 1. This is a marker for systemic inflammation. So mostly in high-fat diet models, be it rodent and non-mammalian, even in obese and diabetic subjects as well, plasma MCP1 are elevated just because one, there's a chronic state of inflammation perpetuating in their system because of the metabolic aberrations that they're going through because they're obese. And so it's challenging enough because you could imagine it's not your system itself is continuously going into breaking down your fat deposits, and so you have high amounts of lipids floating in your blood. At the same time, there's a lot of glucose because at that point where you become diabetic, your liver keeps generating a lot more glucose for gluconeogenesis. And so there's so much assault going on, and so you cannot help but be inflamed. And so for this particular one, our seaweed supplementation significantly reduced the amount of plasma MCP1 in our high-fat fed group. And so that is very promising. That's why I'm so invested in this part. And I'm still working on it at this point. Yeah. And then here I'm showing you a PCA plot. This is for the beta diversity index for my gut microbiome. So as I mentioned earlier, the idea is when you have a metabolic assault, say for example, a high-fat diet, high-fat fed mouse, you could see how different the gut microbiome profile would be from a low-fat fed and a high-fat fed. So here, this is my high-fat fed group, and this is the low-fat fed group. So these are individual mouse, and so we have a different plot for major genus that were detected in my fecal samples. So essentially, we caged our mouse individually, and we collected feces thereof. And this is the high-fat with the duals. And so you could imagine, ideally, you would expect that this would overlap with the high fat, but you can see here that at some point, the supplementation of the seaweed did averted the development of the phenotypical gut microbiome associated with high fat, and our high fat fed group with the seaweed somehow is closer to the low fat, suggesting that there was some degree of rescue. And so in this, you could look at three potential angles. That our seaweed has anti-diabetic potential, it has anti-inflammatory as well as a protective effect in the gut. And so this is more of my pitch, say when like we're developing some proposals. And so we're expecting something hopefully to go further from this point. Yeah, interrogation approach in vitro. So as I mentioned earlier, we can look into in vitro if in vivo is not possible because it's significantly expensive. And so say for example, for this one, this is 3B3L1. This is a adipocyte. So as I mentioned earlier, in the context of obesity, you have a high amount of fat pads. Your fat pads are constituted with adipocytes. These are cells, fat cells. And so the idea is like on a normal growing cell, they're like fibroblastic. But over time, they accumulate fats. And so you can see these blobs. These are actually fat droplets. And so this is a good screening approach if you're looking for any bioactive component that could potentially halt the development and accumulation of fat droplets in your cells, meaning it could have an anti-adipogenic or anti-lipogenic effects. So meaning that could suppress growth of more adipocytes, so meaning you don't have more bags for your fat deposits, or two, you could have less lipids, and so you would have less problem in lipogenesis, in lipolytic breakdown eventually. So this has been a method that they did in the screening for anti-obesity articles from Callirpa. And so they did that in vitro using the cell model, and then they did that in vivo, and they saw that some of the polyphenols derived from Callirpa were able to protect the mouse model from obesity after supplementation. Another approach is using this macrophages. This is raw 264.7. This is the most used cell line that I'm working right now. I'm working in anti-inflammatory screens. So essentially because they are macrophages, when you stimulate them with lipopolysaccharide, they produce nitric oxide because they have inducible nitric oxide synthase. And so in the process of production of nitric oxide, you can look at whether your test article would be able to reduce the amount of nitric oxide produced in the system, at least without killing the cell, compared to your anti-inflammatory control, say dexamethasone. So that's essentially a screening approach. And then from there, if you do have a positive, you can go through forward in like upscaling your cultures and look into gene and protein expression. Another cell line that I'm working with is a liver cancer cell. This is from human. And so HepG2 is quite like a common model for oxidative stress. And so, as I've mentioned, usually in the context of obesity, you don't only have inflammation. Usually inflammation arises because there's big oxidative stress happening. So it's a good screen for that. So you can use this cell line as well. And then CACO2 is a colon cancer cell. Essentially, the original plan that I have for this one was to use this in a trans well. So essentially, it would simulate more like a lumen, since like you have your gut, and so it develops villi, and so it could simulate how whatever bioactive compound you're putting in, how it would go and cross the gut. And so that would be ideal in trying to answer whether whatever test sample you're putting in your cell model, if taken in by a person orally, could it actually get into the blood? Because you know that the gut condition would not necessarily be favorable. So that's a good measure of bioavailability. However, since I'm not yet on that part of my study, what I'm using this right now is for a cheaper source of my DPP4 enzyme. So since this is from human, so one, I'm working with DPP4 from a human cell. Because you could imagine how costly a purified DPP4 enzyme is if you're doing a routine screen. And so what I've been doing is I'm growing them in 96 well plates, and I'm doing my DPP4 screen there using a cell-based model. A differentiated DPP4, at least, so they grow confluent and they grow this structure, they actually express DPP4 at least after 5 days. And so it gives me like a cheaper DPP4 source. And so that's what I'm using for my screens. Now, I place this as an interrogation approach because essentially the are you still hearing me? Okay.
Yes.
Okay, I'm sorry. I was freaked out that I felt like I lost everyone. Did I lost any slides?
Wala po ate. Wala po ate, Rufa. On those, BAPS fractionation.
Okay, good. I'm sorry. I because like everybody quite quiet. So part of the dose BAP fractionation is that I wanted to interrogate what part of my seaweed really is doing what. And so we are fractionating parts of it. And so you can see here, so I appreciate that Sir Brian did a good job in walking through the process for proteomics. So essentially here, you can see this is a splitter. So I'm collecting my fractions. This is from an LC going here, and this is a mass spec. So what I'm doing is I'm collecting fraction at the same time, getting a fingerprint of whatever I am eluting. So that kind of like saves time, I guess. But yeah, because this is an older mass spec, so I think I have like more free hand in using this. So yeah, that was helpful. So in getting this fraction, okay, so here I'm providing just a chromatogram of one of the samples that I got. And so I eluted this over 100 minute and then collected 2-minute fractions and then pulled this and did my cell screen. So imagine the cells that I showed earlier. I was using here 264.7, the raw cells, the macrophages that I stimulated. And so here you could see that it's only 5A that significantly reduced nitric oxide in my cells, showing that this could have the anti-inflammatory factor that I'm looking at. And so, but this is pooled samples. So I'm trying to interrogate which component in my D5A is that. So I would be sub-fractionating from there. However, the limit with this is that these are small samples. And so I guess if you want to go further, make sure you have a big amount. Like when you do screening, you'd be surprised how much sample you go through. So yeah, that's part of the interrogation approach that I would recommend. Another approach is the in silico. So as Milo mentioned earlier, I am working with seaweed, and so I did a lot of extraction optimization, some isolation approach, and then we worked down with the hydrolysis preparation, and then bioactivity screens. And the problem there is that there's so many potential that you could evaluate, but you don't have a cell line, you don't have the reagent. So you could explore in silico in that. And so here I'm showing you, I have here 2, 4, 6, 7. So this labeled columns here, these are actually the hydrolysis that went through. So the original sample set that I was screening about 144 samples and then cut them down to 50 and then cut them down until they became 7. And from here, you could see that I listed out anti-inflammatory peptides, DPP4, antihypertensive. So these are all in silico tested. So I will walk you through how we did this. So in the peptide identification, Sir Brian mentioned earlier, we did de novo sequencing because the protein for the seaweed that I'm looking at is not well annotated in UniProt. And so we ended up using de novo for that. And so with the screens that I've been doing, so these 7 hydrolysis that I've been working have positive hits in DPP4, inflammation screen, and antioxidant and H1. And so I kind of pulled them together because some of the bioactive peptides potentially could be multifunctional. And so it's preferred that one bioactive peptide would have multiple applications so you could explore multiple arms in the process. And so in this hits, I developed my protein database, which consists of 7 species of annotated proteins from same family at least. And then I have 842 proteins in that database. And then from there, I had to just cross-check the peptides that I have versus the database. And so the idea is the peptides that I have would be considered as seaweed peptides. And using those, I did my bioactivity prospecting. So for the anti-inflammatory peptides, I used this platform, it's pre-AFP. I used IBTP4 SEM. This is for the DPP4 inhibitor screen. And then I used AHT pin for antihypertensive peptide screen. And so essentially you put in here a FASTA format, and then you just have to set specific thresholds that would be applicable in your specific context. Also, part of the in silico work, you can check on stability, toxicity, and bitterness. Particularly, I am interested with peptide stability because one, as I mentioned, you do have challenging condition in your stomach and in your gut. And you have to make sure that if you would want your bioactive test article to be still active once taken orally, you have to make sure that it goes through that intestinal lining and gets into your blood. And so you have to make sure that it's stable at that point. And so the half-life in the intestine-like environment, this is an in silico platform, and as well as your half-life in the plasma. So you could do like a predictive modeling for that, and you can use these platforms as well. I have provided here in yellow the papers that reported this platforms. You could also work with the toxicity prediction because imagine there, as I mentioned, the kahala halide, it's a dipeptide. So you could imagine that's an anti-tumor. So essentially how an anti-tumor peptide works is it kills a tumor cell. So you could imagine that some of the peptides could kill a cancer cell, but at the same time kill other cells in the process. So you have to filter through the non-toxic ones if you're going to go forward with a bioactive peptide as a supplement or something. And then since I'm looking at a food application, so I have to make sure that it's not bitter. And so for bitterness prediction, you can explore this platform, the Ibeater. And another aspect to this was not part of a published work, but we are exploring these platforms. These are specific for small molecules, but essentially, if you look at it from the standpoint of any drugs or even like for dietary purposes, you can imagine that food is being consumed, and so you have to look at its metabolic phase from the standpoint of absorption, like how it is being absorbed by your body, how it is being distributed around your body, but at the same time metabolized and excreted. And so there's a predictive platform for that. So you can explore PKCSM, or you could check AdNet Lab for that. They have been very, I would say, they are helpful very much, but there are very numerous caveats that you have to be particular about. And I would recommend that somebody who has prior work into it would do this side by side, because we're working with signature structures that would be identifiers for how they would be reacting, say, for example, in your absorption or in a metabolic condition. So yeah, anyway, for the application for those platforms, we did publish a paper in 21 covering those platforms. And so you could see, I provided here the link and the predictive purpose for those platforms that I provided earlier. So why go through the process with that? As I've mentioned, one, it's cheaper. Two, you could not screen for all those things in the lab. That would be super costly. And three, you're covering your grounds. Your chances of catching fish is higher. And so that's why we could explore, we could start like looking at our options. You can go in vivo, in vitro, or maybe in silico. So the question now, where can we begin? At this point, like we see that there's a big potential. We can, I guess, explore those options for the interrogation strategies. I guess at this point, we can at least strengthen ourselves with literatures. This paper is actually a good start. Like it covers mainly what is known out there for seaweeds as nutraceuticals. The drug research is very competitive. It's almost very challenging to find our niche there, but the nutraceuticals market is quite open. Japan has what they call hoshu, so it's like food for specific health use. It's technically not a drug, but you have to have claims to it at some point that it could improve, say, for example, hypertension, but it's technically not a drug. So there's a niche for that, and the market for nutraceuticals is expected to grow at least 40% in the next 5 years. And so that would be very promising as to us exploring that. But as a research community, how do we begin from where we're at right now? I think it would be very strategic for us to have a solid research initiative that's based on functional food. So these are some of the research projects, initiatives across Europe that has been funded, and they have been very productive for at least the last 15 years. They just have seed funds, and then that kept rolling. And so particularly SWAFOX, SWAFOX is the project that tried to elucidate the effect of polyphenols on inflammatory markers in humans. And so they did a clinical trial for that. And they also looked into how these polyphenols would be handled, so the metabolic fate of the polyphenols. And so it was one of the earliest works that show the biotransformation of chlorotannins and how the more active ones would be the breakdown products instead of the original product that you actually take in. So those are some of the strategies we can begin with, and it would be very helpful. I think I've seen NFRDI people, and this is a shout out for you. I hope we can have some research initiatives for this. Another example would be the Nutrimara. So I just provided here how it's established, who supports it, and the program itself. So this is how they are working with stakeholder groups. So they have made this mainly Ireland based. So the lab that I'm really closely following with has been doing a lot of seaweed work, and this is in Ireland. And so they have a very close knit relationship with the industry, the academic and research institutes, with the government. And so one, not only are the people trained, the little tiny researches for undergrads can be used as preliminary work, and we can build on from there. And I think that's one thing that we're not really maximizing. Yes, we're screening, but it's totally separate. And so we don't even have a solid library of what's already done. And so some people could have been replicating the same thing and finding the same thing, and when this person graduates, it's gone. And I think it would be very strategic to have a collection of this so we can explore what has been tested. And so if this did not work, what other things we can explore? Because I think it's very advantageous for Philippines since we're an archipelago. And two, there's so many things that we can explore. And so I think it would be a good start to have this setup in place. And then two, we can just start in vitro assessment. So this is a 2013 paper, and this same people right now already did one clinical trial where the same seaweed they supplemented on bread for anti-inflammatory effect. And that was published in 2020. So in 7 years, they did a prior work, and then they did reach until clinical trial for 7 years. They also found some of the antihypertensive peptides, and so they're working on into that using a different animal model. So let's just start groundwork, collating our groundwork, our efforts, and then pouring into that whatever has the most promising therapeutic potential. We can do in silico. So in silico prediction and Rufa has been done in 2017. That has been published, so you can access that work. It's basically just mapping what could be possibly there. And then for some of the screens that we can easily do, we can do a lot of the enzyme inhibitions. I guess like for identification, we could collaborate for bigger labs that has chromatographic identification capability. The proteomics, I heard there's a lab that's being set up, so that's promising in UPV. But we can begin from there. We can tap from there. From the standpoint of NVivo models, so if we do the groundwork for in vitro and in silico, we can go for NVivo. And this paper was published in 2018. They used a different model, not a mammalian model. They used zebrafish. And so the same idea, you induce your animal for obesity and then look how supplementation would affect phenotypical development. And so in this case, you could see how different they are body-wise. And so we can explore for cheaper animal models. And I know Japan has a good amount of zebrafish, and I guess we can tap our contacts from there. I think that's a good way to go. And should we have hits from there, by then we can look into mammalian models. And of course, you can look into mouse, because normally that's how preclinical trials usually conclude, like what is the effect on the mouse. But from there, we can ask bigger labs that have the capability to do this on our behalf, not because we're lesser people, but just because we're good at the groundwork. And so that collaboration could strengthen niche, us putting in a solid therapeutic potential assessment platform, like a pipeline that would narrow down which things we could move forward from there on. And so it would, one, strengthen our expertise and our strengths. We do have the resource. We do have a lot of willing people who would do a lot of the screening, the groundwork, and then do just like a cheaper animal model and then scale from there. That would be a good place to start. And so at the end of the day, ultimately, why do we have to sweat it all out in all this? You get to look at the bigger picture. Philippines has a lot of seaweed, and across Asia, there's even a higher diversity available for us. And so if we start in our own backyard, you could imagine how much potential we are tapping, not only for metabolic health improvement strategy, just you directly eating it as a component in your diet, but also from nutraceuticals and direct therapeutics. And so the market application for that is beyond imagination right now. We're talking about $1 billion. And so there's so much promise to it. But other than these reasons, maybe you have more complex ones. And so that's the challenge that I would pose to everyone around. This is my project that I feel like passionate about, because like if somebody asked me about seaweed, like, oh yeah, you know what? And we start like talking for hours about seaweed. And so I think there is much to be tapped. And so one just has to be willing to do the groundwork. So yeah, at the end of the day, I would like to challenge everyone that just showing up every day and being willing to answer your questions, or even cast your first questions, like being there to ask questions like what could possibly be done. I think that's something that we have to be willing to sweat out and be keen in observing what is before us. There's just so much that could impress you if you are keen enough to observe. And you'll be surprised. There's a lot of things to marvel about. So today, I hope I did give a fair amount of explanation as to what are the research directions there is and that can be explored in seaweed for health applications. So with that, I'd like to acknowledge the Corn Lab, the Tin Corvallis in Astoria, the Linus Pauling Center, where I'm currently housed, the Food Science and the Seafood Lab. I do get my seaweed from the Hatfield Marine Science Center, and the cool part of our Seafood Lab as well is that I'm part of the Coastal Oregon Marine Science Research Center consortia, some sort, and so it feels more like a College of Fisheries. So it feels like home. And the Fulbright with the Philippine American Education Foundation, SHED, and of course my home institutions, Zamboanga State College of Marine Science and Technology. And so with that, I think I did hit my target time. Thank you very much for listening. And I think now I'm ready for your questions.